Okay. Yeah, good afternoon, everyone. I'm Ruoshi, and today I'm going to present my recent work called Spacedust for de novo discovery of conserved gene clusters in microbial genomes. I'll start by highlighting that the fact that functional potential encoded in the microbiomes, such as the human gut, soil, or ocean, remains poorly understood. In the human human gut microbiome, for example, only 60% of the protein coding genes may be functionally annotated. Traditionally, function annotation is by using a homology inference, that is sequence similarity search. However, sequence alignment is challenging in the sense to find evolutionarily distant homologs. And in many cases, function can be even conserved with very low sequence identity, below 20%. Take this following example of ubiquitin and the SUMO, small ubiquitin-like modifier. They only share a sequence identity of 16%, although they still have very similar structure and function. And protein structures are typically more conserved than sequences. For the same two proteins, we can use a tool called FOTSeq. That is a tool recently developed in our group to align their structures. FOTSeq discretizes structures into sequences over a structural alphabet called 3DI. And in the same cases, you could see that with the FOTSeq alignment, the sequence identity is much more pronounced. Hence, you could derive a homology based on that. And one thing to add is that FOTSeq is extremely fast. It's 4 to 5 magnitudes faster than the state-of-the-art structure alignment method. So this makes structure alignment for a large data set possible. This also includes the fact that the recent advancements in the protein structure prediction, like AlphaFold and ESMFold. So the predicted protein structures with near experimental accuracy are becoming available at the scale of a known protein universe. So since the release of AlphaFold database, there are over 200 million protein structures now deposited in the database. And beyond homology, the function can be further complemented by a gene neighborhood conservation. So it was observed that gene organization does not appear random, regardless in prokaryotes, usually organized into operons, and further in eukaryotes like fungi and plants. So there are also functionally related genes are normally organized in the proximity of each other. However, on the larger scale, gene neighborhood is usually not conserved unless there is a selection pressure to retain them in place. So many tools, like we have seen earlier, detect specific types of gene clusters, such as BGCs, or can search for known cluster families if you have already a set of clusters. But today we present Spacedust. It's a tool for systematic de novo discovery of conserved gene clusters in large-scale genome datasets. And Spacedust mainly consists of two parts. So the first one is homology search, powered by the sensitive structure search using FOTSeq. And the second is a cluster detection for conserved gene neighborhood. So Spacedust detects all instances of homologous clusters of conserved gene neighborhood, and without relying on any prior information. Thus, it is a general method and also de novo. So I'll go into a bit more details of how Spacedust work. So first, we take nucleotide sequences from genomes and contigs. Using Prodigo to extract the protein coding genes and their coordinates. And next, we would map the proteins with a very high sequence similarity cutoff to structured DB, for example, AlphaFold DB. And the mapped structures are then searched using FOLDSeq, and there are a small fractions of unmapped sequences. We will still use MM6 to do the homology search. And next, we would filter all the homologous hits using their pairwise e-values, and also further group them by the by each genome pair. So next, we move to the cluster detection. We detect clusters of these filtered hits per pair of genomes. And in details, how are these cluster detected? So we can imagine we can imagine all the hits are aligned on a grid of the size of genome query and genome target. And then we would look for significant gene neighborhood conservation. And for any clusters of hits, we would check using two p-values called clustering p-value, which is the probability of seeing two hits, for example, here within the square of a size of 2, and the ordering p-value, which means the pair of genes that have conserved gene order and strength. In this case, this is 1. And then we would not consider cases with too big gap. Here we define this gap D as a 2. And we would compute a cluster match score using the sum of negative logarithm of the two p-values. And then in each iteration, we would merge the hit with highest score. So the next step here would be try to cluster either the pink gene or the green gene. And in this case, the clustering p-value would still be the same because the size of the square would be the same. But the pink one has a slightly worse ordering p-value. Therefore, we would take the green. And simultaneously, we annotate the proteins using eggnog mapper, which provides comprehensive functional information to complement the clusters. So at last, we would output all the clusters in tabular format. So we applied Spacedust on a dataset of diverse bacteria of a size of 1.3 K, which was clustered from 7,000 complete bacterial genomes extracted from the KEGG collection. And we first clustered the KEGG collection genomes with a 70% amino acid identity, which roughly corresponds to genus level clustering. And then we take the 1.3K representative genomes, which map to 3.8 million protein structures. And we did it all against all comparison using Spacedust. And this run completed in around 72 hours for the homology search and around 2 hours for the cluster detection on 2 servers with 180 cores each. And this gave us 90.5 million pairwise clusters. And so now that we have so many pairwise clusters, how do we go about to visualize? So in order to get a better view of these clusters, we decided to visualize the pairwise clusters for each genomes against the full dataset on the interactive presence-absence heat map. So I show here an example of cyanobacteria from this 1.3K dataset. And the proteins from the query are arranged by their nucleotide coordinates on the genome. And then the y-axis are the 1.3K reference genomes. So for better visibility, we could zoom into one of these regions. And there we can see the presence of conserved gene clusters across all the reference database. And I show now in 3. Can I use the? Can you see my cursor? Yeah. So 3 cyanobacteria specific clusters. And we could, so with the eggnog mapper, we could assign functions to all the genes. And for these 3 clusters, they are involved in photosynthesis or mobility. And next, I want to prove that the pairwise clusters recovered from Spacedust also can have so more specialized functions. So I compared the results to the antiviral defense systems that annotated using another specialized tool called Padlock. And we see that the Spacedust could recover majority of the antiviral defense systems. And I show now an example here, the blue arrow pointing to this arm, another antiviral defense system. And so here you could see the cluster boundaries in using the gray shadings, and also the genomic context can be easily plotted. And Spacedust can also be applied to expand cluster families that are well known. And for this example, we searched the genes from a novel CRISPR-Cas defense system called CRISPR subtype 3E that is characterized by a single protein effector known as Cas711. And we searched this query defense system locus against the GTDB. And from the GTDB, we would have identified 7 new instances of the previously unreported CRISPR subtype 3E. And to conclude, I hope I convinced you that Spacedust is a general method that is not limited to finding any specific types of clusters. And it is sensitive to find distant homologs by leveraging fold-seek's structure search. And we proved that it is fast enough to support all versus all searches at the scale of thousands of genomes. And with the integration of the eggnog mapper, we could facilitate function deductions of the function members, the cluster members. And we are currently developing a a web server for visualizing this pre-generated cluster set or any user-provided input. Furthermore, the Spacedust is also applied on unsupervised systematic operon detection for all bacterial genomes that will be presented by Hong on Wednesday. So I will not go very much into details, but I can just say that we use the conserved cluster matches by Spacedust plus some other constraints for operons, and we could reach a very high accuracy. So please go to her talk if you're interested. And with this, I can, and we also have stickers later at the poster session. Yeah, feel free to come and claim them. I thank everyone who's involved in this project, and especially the Zoding Lab members, and I can take your questions.
Great. I think we're still ahead of time, so we do have time for questions.
Thanks. Nice talk. I just have a question regarding your cluster annotation. So you are annotating, like I might have missed this thing. So you annotate each member in a cluster, or you assign a function to that cluster?
So currently, we only assign function to each member of the cluster. We don't generalize what this cluster does. But I think for if you want to annotate unknown genes, then maybe knowing the function or generalizing the function from the rest of the members would help.
And so I don't know, like, what will be your approach? Like if the cluster member, you have 3 members in a cluster, 2 of them get one function and 1 get a different function. So if we want to assign a function to that cluster, then how you will proceed? Like
usually like the functions so what we see from the data set is mostly protein complexes or metabolic clusters that we already know. So so if you have something outside of that, yeah, it's a bit difficult because it's all text level generalization. Yeah, so currently there is no good way of summarizing what a cluster does. Yeah.